All right, biologists, let's take a look here at translation. Okay, this is part of subtopic 2.7, DNA replication, transcription, and translation. And the IB tells you that translation is the synthesis of polypeptides on ribosomes. Now we can replace the word polypeptides for the word proteins. Right, so translation is the synthesis of proteins. Who makes those proteins? The ribosome. Quick review. First, you had the DNA, and the DNA has the recipe, has the code for all proteins. From DNA, you make a copy of RNA, and this process is called transcription. And then from the message in the RNA, you have the protein. And this process of translating the nucleic acid language, the language of RNA, into the amino acid language, the language of proteins, this process is called translation. So we have our friend here, the messenger RNA, and the messenger RNA has the recipe for the protein who's gonna cook this recipe. The ribosome here in green, the ribosome is made of two parts, the large subunit and the small subunit. The ribosome is going to assemble the two subunits are going to come together, forming kind of this mRNA sandwich here with the large subunit on one side and the small subunit in the other. It is also good to remember that prokaryotes have what we call 70S ribosome, and eukaryotes have 80S ribosomes. What does that mean? It only means that prokaryotes like archaea and bacteria have a lighter ribosome than eukaryotes like animals and fungi, plants, and protists. That's the only difference. Otherwise, they work exactly the same. They do the same things in the same manner. So the ribosome is going to read a group of 3 bases here in the mRNA sequence, and this group of 3 bases is called codon. Each codon represents one amino acid, and the amino acid is brought by this guy here. This guy is the tRNA. T stands for transport because they are transporting the amino acid. Now, if you are higher level biology, you are going to remember that the large subunit has 3 sites. If you're standard level, don't worry about this. But higher level, you're going to remember there is a site A, a site P, and it's not drawn here, but there is a site E, right? You have the site A, site P, and site E. A is where the amino acid arrives. The amino acid arrives, so A for arrival. Then the peptide bond is formed on the P side, P for peptide. So the peptide bond is this covalent bond between 2 amino acids. It's a condensation reaction. And then the tRNA now leaves the ribosome on the E side, the exit site. A arrives, P, peptide bond, E, exit of tRNAs, and here is our peptide being built. All right, as we said, codons of 3 bases on mRNA correspond to 1 amino acid and a peptide. This table here is kind of like the cheat code that the ribosome has. So the ribosome has this table, the ribosome knows this, the ribosome uses this to know what amino acid the codon represents. The table that tells you what codon the amino acid represents or what amino acid goes with this codon is called genetic code. The genetic code is a table that correlates every codon to every amino acid. And this is important because you have only 4 letters, 4 different bases, but you have 20 amino acids. So how do you correspond amino acids to bases? Well, if you had only 1 base, if you read only 1 base at a time, you can only represent 4 things because you have 4 bases. Now, if you represent with 2 bases, well, now you have 16 possible combinations. That's better, but still not enough. However, if you represent with 3 bases, now you have 64 combinations. So you have 64 different codons for your 20 amino acids. You have more codons than amino acids, which allows this thing here. The genetic code is degenerate. The genetic code is degenerate, which means more than one codon can represent the same amino acid. More than one codon can represent the same amino acid. Let's take a look at the codons. A, U, and A, C. I'll take a look here at the first position. The first position is A, right? So here, we're going to take a look at this line here. The second position is A. So here we have the second position is A. So we're taking a look at these codons here, sorry, these amino acids here. Now the third position is U, and the amino acid is ASN. Do you need to know the name of the amino acid? No. So just write ASN, you're good. Now, what if the third position is a C? Well, you have the C here, and it's still the same amino acid. This is what degenerate means. 2 codons are representing the same amino acid, and that's fine. That gives you extra protection in case of mutation, because what if your mRNA is supposed to be AAU, but a mutation in the DNA causes it to be AAC? There will be no change in the amino acid sequence, and this mutation is silent. So being degenerate is a good thing. More than one codon represents the same amino acid, and that's good. The other thing we notice is that the genetic code is universal. That means all species, virtually all species, will use the same table, which means I can take a human gene and put in a bacteria, and the bacteria will make the human protein. That means I can take a jellyfish gene and put in a regular fish, and then you have a fish with jellyfish protein. You can have a bacteria gene in corn, and you have a corn that makes a bacterial protein. That works because the genetic code is universal. The relationship between codons and amino acids is the same for virtually all species.